Hello, hopefully you can hear me and hopefully you've been not waiting too long for this to get started. So this is going to be the first session that we do on carbohydrates. So fingers crossed you are a year 12 or a year 13 student and you decided to do your A-levels in the last couple of years and you've hopefully just found SnapRevise and you've hopefully found all of these wonderful things that we're offering you at the moment. It's really cool to see all of your comments. So I've got all your comments loading up down the side of my screen right now. And it's pretty cool to see that so many of you are so excited about this. I saw someone earlier said that they've been literally waiting for this all day, which is slightly bad for your teachers, but apart from that, I think that's quite a good thing. So what I'm going to do is I am going to start sharing my screen with you guys so you can see what I am doing. Here we go. And then we shall get started if I put this back to the right place. Ooh. There we go. How's that looking? Perfect. I think that's looking great. So yeah, welcome to SnapRevise. Hopefully your year has been going quite well so far. Hopefully after, well, for a couple of weeks, it's not been too bad getting back into the swing of things. This is going to be the very first session. So we're looking at carbohydrates today, which tends to be where most biology specifications tend to begin. Okay. Hello to everyone who is constantly saying hi to me at the moment. That is nice. Okay, so just in case you want to know a little bit more about me, which I'm sure you're all dying to find out, I'm the head of biology here at SnapRevise. You should be able to see the logo somewhere behind me. I've got a bachelor's degree in biology, so I studied my degree about 5 years-ish ago. I went to the University of Southampton and I did my degree in just pure biology, and I realized that it is the best subject in the world, much better than chemistry and physics, but it is the best. So I became a teacher, and for the last 4 years, I've been working in a school, or a couple of schools actually, where I've been teaching A-level biology as the lead teacher. So I know quite a lot. I think I know what I'm talking about at this stage, fingers crossed. Okay, let's get started then. So in case you were wondering what these web classes were, I saw some comments basically saying, oh, how long is this going to go on for? And I think someone from SnapRevise hopefully answered your question about that. So these are going to be 45-minute sessions, and you should be able to find this PowerPoint. So if you have a look at this PowerPoint, if you look at the messages on YouTube right now, you should see that there is a blank presentation that you guys can start to fill in. So in preparation for university, really, if you have a look at that, you can download it, you can write notes on it, you can download it somewhere, and you can read through it again and again and again if you really, really want to. Let's see, many of you finding that? Oh, there it is. Hopefully you've just seen that pop up. Biology is better than chemistry, whoever that was. Anyway, I am really biased. Just so you know what we're going to be talking about then, the whole point of SnapRevise isn't just to be instead of your lessons that you're having at school. Hopefully this is going to be a recap, so it's you'll see some things that you've hopefully seen before. I'm going to go over the key points. So there are some bits that I might not go over, which I'll tell you exactly what that will be for this session. There are some key points that I won't go over. And after I've done that, we'll go over some exam technique, we'll look at some questions. And then as someone said this earlier, I'll be here for about an hour. So if there's any questions at the end of these 15 minutes, feel free to just post them and I'll do my best to get back to you. I'll try and answer it over the website. Okay, just so you know as well, as you've probably worked out, this is free. So for now, this is free. For the next 4 weeks or so, you've got the weekly sessions, you've got me who will be doing this, and you've got one of my colleagues, the head of chemistry, who's going to be doing some chemistry sessions. But up until October the 16th, I think you've got this. Okay, I'm going to get started. So just so you know as well, just to keep you here, there's got to be an incentive, doesn't there? We've got a very big discount code for SnapRevise at the end of this session, so feel free to stay tuned for that. It'll be the very last slide. Okay, right. What I want to do first is whenever I'm teaching, I tend to like to start by looking at what you guys know, so I know roughly where to be pitching this at. So I have made something that I call a pyramid of success. So if you start at the very, very bottom and work your way up, there are some questions there which I'd like you to have a go at doing. If you post them, I'll tell you if you're right in a second. Okay, so have a look at some of these questions, see if you know the answers. I'll keep an eye on my little messages and see if you are guessing this. Okay, I'll give you probably about 30 seconds and then I'll start talking again. So I'll keep having a look. Austin McCullen, someone is here. Ooh. I'm seeing some right answers so far. This is good. Although, is it worth thinking, are all sugars glucose? Nice, this is looking good. Glucose, monosaccharides, disaccharides. Okay, so in terms of scientific name for sugars, we tend to call them carbohydrates. So lots of you are saying things like maltose. I just saw maltose, two glucose molecules, which have had a condensation reaction happening. Starch is a polysaccharide, which is made of loads of glucose molecules. The scientific name we tend to use for sugars tends to be carbohydrates. What about this next one? So what is the name for lots of sugars linked together? How many people are getting that right? Ribose, deoxyribose, they are sugars too. I'm seeing some people saying some polysaccharides. That's nice. What else are we saying? DNA. DNA isn't quite lots of sugars linked together. It has sugars in it, but it also has lots of phosphates. It has inorganic bases as well. Lovely. I'm seeing loads of people saying polysaccharides. I like that. You are spot on. Lots of sugars linked together is a polysaccharide. What kind of organism produces sugars? It's always one answer that tends to come up here from my experience that I've had. Who can tell me the organisms that produce sugars? Starch and glycogen, looking at, yeah, some big molecules. Nice. Excellent. Right, that's what I expected you to come up with. So lots of you, in case you're having a look, lots of you are saying plants right now. There are more than just plants. There is something else too, or there's another organism I like to think of. Animals and plants. I can think of one animal that can do this, but it's a very much in the minority. Definitely not all organisms can produce their own sugars. Algae, yes, someone has just said the right thing. Algae is totally right. Bacteria too. There is a bacteria called cyanobacteria, which can also make its own sugars. Humans, no. Autotroph, lovely, great word, autotroph, that is definitely right. Animals, I think it's some form of sea slug, the only one I could think of. Nice, looking good. I like the whoop whoop. Need more of them. Okay, what is a balanced symbol equation for photosynthesis? Who can come up with that? Who's gonna be the quickest? Ooh. So far, no one. Uh-oh. Algae aren't plants. So algae are a different type of organism. So algae are a protist for Robert Goddard, who just said that. Lovely. So what we got carbon dioxide plus water, plus some energy, lovely, in the form of light, makes C6H12O6, glucose, or any sugar really, and oxygen. Lovely, that is looking good. An autotroph is just any organism that can make its own food, so the opposite of a heterotroph. Not balanced though. Yeah, good point. That isn't balanced. It needs to have 666 before the carbon dioxide, water, and oxygen. I've always remembered that as the devil's number. Lovely. Someone is getting way ahead of themselves, looking at the light-dependent and independent reactions. I will be talking about that for my next session, which is on Wednesday. Lovely. You guys are smashing this. What is the process by which sugar is used by living organisms? I've seen one person say it. Yes, lovely. Vanessa's got it as well. Respiration is the process by which sugar is used. Finally, I like to finish with something about, can you tell me a few other interesting facts? Any interesting facts about carbohydrates? Go on, I'll give you a few seconds to come up with something. Just lots of respirations. Way. Nice. Any other facts? Bread. Yes, bread is carbohydrates. Lovely. Carbon, hydrogen, nitrogen, and oxygen. Ooh. So Molly Brown, not entirely sure I agree with you there. Carbohydrates contain carbon, hydrogen, and oxygen. They don't contain nitrogen. Optical isomers, love it. Someone is getting ahead of themselves again. Nice. Energy stores. Reducing sugars. Oh, guys, this is amazing. Clearly you have been listening in your lessons. This is something which I wish all of my students were like sometimes. Right, I'm going to move on. Someone has just said yummy. That is excellent. And someone said they don't release as much energy as lipids. I like that. Okay, so let's look at what we're going to do today. I'm going to stop looking at the comments for just now. So by the end of this session then, I need for you guys to be able to tell me what the difference is between monomers and polymers. Nice, easy start. I want you to be able to tell me what alpha glucose is and its comparison with beta glucose. Again, fairly straightforward. And then finally, we're going to look at something a little more difficult, a little more biological in my mind, how the structure of cellulose and starch relate to their function. So this is always something that biologists like to think about. How does the structure of something relate to its function? Okay, that is consistently what biologists are trying to find out. If you know anything about DNA, that's exactly what the pioneers of DNA were looking for. Just some things that I won't cover. So I am not going to cover these two bad boys here. So I am not going to cover sucrose and lactose. This is really, really straightforward, what sucrose and lactose are made from. I just don't want to cover it because I want to get into the key points. Okay, you'll probably see a comment coming up in a second, which will show you a link for how sucrose and lactose are made, which is one of our other videos that we've made. Okay, I'm also not going to look at the food test for carbohydrates. I'm going to do another session on that in the future, so I'm going to ignore that. And finally, because some of you guys are doing some exam boards that don't cover pentose sugars, I'm not going to bother with that. It's just the one that does. I think it's one of the EdXL. I think it's the Salters one. So I'm not going to bother with that because it'd be a waste of the rest of your time. Okay, so that's what we're going to be looking at. Here are the specification points. This is more for your own reference. So this is basically what I've always told my students to do. Read through this. If you can do it all, smash in. If you can't, then that's something you need to be looking at. Just so you see, look, here are some of these biochemical tests down at the bottom. There's some more up here. There you go. I wasn't going to talk about that either. Again, OCR, same sort of thing. Someone who mentioned carbon, hydrogen, oxygen, and carbohydrates earlier. There it is. There are the EdXL ones. We have two EdXL ones that we need to be looking at. I can't remember where I had it. Somewhere on here, you'll probably, I'm sure one of you has found it before I have. There's another sugar, which I'm not going to be talking about. It's somewhere on there. I'll let you find it. Okay, so getting started then with the actual learning. God, it's taken a while to get here, hasn't it? We need to firstly know then what the difference is between a monomer and a polymer. Okay, so in terms of what a monomer is, you need to be aware, and this is so important that you use these words. Monomers you need to be able to define as being small, repeating, excuse my handwriting, subunits. Okay, your examiners are looking for you to be able to use terms like repeating. So a monomer is small and it is something which will repeat itself again and again and again. I chose this little picture at the bottom here because you can see these little subunits, right? Each little subunit is making up this bigger molecule. So each of these is a monomer. Collectively, when they are all joined together, we end up with something which is a polymer. Okay, so that is the definition we need for a monomer, small repeating subunits. Conversely, when we look at a polymer, we need to know that they are large. And then the word that you tend to be looking for is complex. Okay, I'm going to keep the word molecules in there too. So they are large, complex molecules. Okay, they are made of monomers. Okay, that is our definition for a polymer. Okay, so you need to keep coming back to this whenever you do any of the sort of biomolecules in biology. We keep coming back to this monomer and polymer. The monomers are repeating. They're the small repeated subunits. The polymers are these much larger things that we build up from those monomers. Okay, this is something as well which tends to be the difference here between getting probably like a C and a D, which I'm sure is not where all of you are aiming for, but this is what lots of people kind of fail at when they first start. Okay, looking at these words and working out what they mean is really, really helpful. It makes your life so much easier. So in terms of the vocab, you guys need to know that this word mono here, think of like a monobrow or something. It just means one. Okay, saccharide, this word saccharide, you might have heard of if you have artificial sweeteners. Saccharide just means sugar, or glucose in this case. So a monosaccharide is one sugar. Disaccharide, something you'll come across, this is two. Polysaccharides, probably heard your maths teacher coming up with terrible jokes about parrots and stuff. Polysaccharides just means many of them. Okay, so our monosaccharide is our one sugar. And you can see a nice diagram of it here. So they always have this hexo sort of shape. You can see how many sides it's got, 1, 2, 3, 4, 5, 6. That is our sugar, that is our single monosaccharide. Our disaccharide here, which I'll talk about in a second, is joined by two of these combined together. Okay, so in terms of our disaccharide, we need to know that they are two monomers bonded together. You tend to lose marks here by not using the right terms. So if you're not using the word bonded, people will say things like joined. You can lose marks for that. And it seems really fussy at the time, but I think it's just the bridge between getting one grade and another. So you need to be using words like bonded. When we start looking at polysaccharides, we need to know that it's just anything more than 2. Okay, the ones that we're going to be looking at are going to be starch in a sec and cellulose. Okay, hopefully so far this has been fairly straightforward. Here's another thing. So this will come up again and 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 again, and you're going to hear it so many times that you're probably going to absolutely loathe it. You guys need to know then that in terms of making our peptides, our disaccharides from our monosaccharides, you have to have something called a condensation reaction. Okay, so condensation reaction because it produces water. So whenever we form, and I'll show you a diagram of this in a second, because I think this is probably one of the most important things from carbohydrates that you need to know. Whenever we are forming a disaccharide, we always make a molecule of water. Okay, so in this case here, I've got one molecule of glucose, another molecule of glucose. They combine to make my disaccharide, and that releases water as well. Okay, I did say I was going to ignore this, but for instance, if I've got a glucose molecule and I add that to another glucose molecule, does anyone know what this forms? What is 2 glucose molecules? I'm going to have another look. Dipeptides is protein. If I said dipeptides again, apologies for that. I meant disaccharides. What do I make if I have 2 glucose molecules? Does anyone know? And whoever just said starch is made from a mixture of amylose and amylopectin. Yes, that's true. Lovely, sweet, guys. This is lovely to see. Glucose plus glucose. There you go. There's a nice little arrow. That makes something called maltose. Hence why Maltesers have that name. So glucose and glucose makes maltose. This is going to release a whole load of water. I've seen exam questions where they've asked you to show the chemical reaction between 2 glucose molecules, and half my class forgot to mention water. Despite all of them knowing that condensation reactions release water, about half of them forgot that. Okay, so a condensation reaction is where you make some water. Okay, this synthesizes. So as opposed to saying making and breaking, you want to start getting in the habit of saying synthesizing. Synthesizing means making. Okay. In terms of the opposite then, if I start off with some polysaccharide, whatever this is, let's say it's starch for argument's sake, in order to break it down, the type of reaction you need to know is called a hydrolysis reaction. Okay. Hydro meaning water, lysis meaning basically breaking apart. Okay, therefore a hydrolysis reaction, when we do them, that is going to be breaking down some water. It's going to use water. So in order for us to be breaking down a molecule, it is going to need to use water. So as a chemical reaction, it looks something like that. You'd have maltose plus water, so plus H2O, I'm going to be lazy. And then we're going to have some form of arrow. That's going to make glucose plus, that's a rubbish plus glucose. Okay, you might be able to tell that if you were trying to do this properly, if you were writing this equation out properly, you'd have it all in a nice sort of linear line. I'm just struggling to fit that in this box. Okay, but you'd want that across a line. So maltose plus H2O goes to glucose plus glucose, or you could say 2 glucose, I guess. Okay, so that's really important. So understanding and remembering the difference of this is really key. This comes up all of the time. So condensation reactions, that is when you are making something, it releases water. The opposite is true of hydrolysis, breaking something down, using water in that process. Okay, I would always think back to this word, look back at your vocab, look at hydro, look at lysis, water splitting. Okie dokie. So this is the part where I think this is the key bit. I think it's absolutely vital. Absolutely every single person watching this video right now can draw a glucose molecule. I think if you can't do this, you're going to really struggle. So in terms of what glucose looks like, I'm going to draw one called alpha glucose for now. Okay, so you've got a carbon here. Carbon joins to another carbon, joins to an oxygen, carbon, 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 lots of carbons. Off of most of these carbons, hang on, I'll draw this one first. So there's an OH down here. There is an OH here. I'm drawing it the other way around. That says HO as opposed to O. because it's the oxygen that always joins to the carbon. That really confused me for a bit when I started doing this. There's a hydrogen here. There's actually another little carbon coming off here. So it's CH2OH. Just so you know, all of these carbons inside, so these two carbons here, they actually have another hydrogen and another OH group, a hydroxyl group joined to them. We just tend not to draw, or I tend to be lazy and don't bother drawing them. So there should be a little H there, should be an OH there. Same with this one, okay? In terms of what a condensation reaction looks like, this is how it works. So I've got another carbon here. So let's draw another glucose molecule. So here's a H, here's an OH, carbon, oxygen, carbon. I'm so sorry about how scruffy this looks. Carbon, carbon, hydrogen, and OH. Right, in terms of seeing, someone just said one full glycosidic bond, love that. In terms of seeing how we're gonna make water, right? Basically what's happening is water, as we know, has the chemical equation H2O. And I'm going to steal 2 hydrogens from here, from my HO and my O. I'm going to steal 2 hydrogens and 1 oxygen. So I'm going to steal these 2 bits here. Okay. You might be able to count, look, there's 1 hydrogen, 2, and an oxygen. Okay. Basically what that does, let's see if I can get this to work. If I can try and rub this out, I might be able to. There's a very good chance. Found the eraser. Love it. Right. If I get rid of that, and that hydrogen, bit of a line there. There you go. So if I get rid of that water, which I've now just made, if I go back to my pen, okay, I now form a bond between this carbon and this oxygen. Okay. Genuinely, the way I've always remembered this is by remembering that this bond that you form has a COC. I remember it as the cock bond. Okay, so here's my carbon, here's my oxygen, here's my carbon. Okay, so as soon as we do a condensation reaction, we're going to make some water. And then this bond here, the COC, remember it however you want. This is called a glycosidic bond. Okay, glyco being a word, if you've done GCSE, you probably came across glycogen and stuff. Glyco just means it's to do with sugar. Okay, so we're looking for this COC. Okay, in terms of my hydrolysis, the opposite is true. So I would draw this again, but I'm actually not going to do it. I'm just going to sort of show you from here what it looks like. In terms of how we do our hydrolysis reaction, we need to break this bond. We need to start to get rid of it. Okay, so the way we do that, let's see if I can show you. So I'm going to start with my H2O. So H2O is going to be really important. And then attached to this carbon here, I'm going to take an O and an H from here, which leaves me with 1 hydrogen left, which is going to join down here. So if I get my eraser up again, I'm so pleased this works. I tried this earlier and I couldn't get the eraser to work, and it was devastating, frankly. So you go away. Lovely. I can take my, what do we have? I can take my OH, add that back down there, and then add my H back there. So that H is connected to that O. It just looks a bit weird because I've drawn a bond this time, whereas it's on that side. So I genuinely think this is so important. If you're going to learn anything from today's little session about carbohydrates, it's this bit here. Okay, if the rest of it is totally beyond you and you haven't done it in lessons yet, fine. This bit is key. You need to know exactly what glucose looks like. You need to be able to draw 2 glucose molecules. There's the other one. And you need to be able to show that, look, to form a glycosidic bond, this bond between sugars, I take this OH here, I've gone Cockney all of a sudden, take that OH there, I combine it with this H here, and you end up having H2O, right? There's my little bond again, the COC. I genuinely would recommend more than anything else, you practice drawing this again and again and again and again until you can do it from memory without even having to think about it, okay? Until you get to that stage, I think it's going to be tricky to progress with lots of biology. I think understanding these bonding models is just so important. It'll make your life easier with lipids, with proteins, with DNA as well. So you might as well try it, okay? Okay, so this is something I like to do, and I'm sure some of your teachers have done this in the past, and it might seem a bit gimmicky, but I genuinely think it's quite important. Before I move on anywhere, I want to see how you guys are kind of feeling with this, because if everyone is totally not understanding it, then I don't see the point in moving on. I think you might as well go back and try and have another look. So if you guys would like to have another look, or if you guys would like to vote, as some of you are doing, see what we get. Lots of ones. I can see one person is super confused. Someone is multiple plus ones. That's not even an option. So don't do that. Someone said, oh, hang on, I want to read that one. Someone said, is something catalyzed by a reaction? Are hydrolysis reactions catalyzed by enzymes, or do you just have to add water? Enzymes are catalysts, as you've just said, right? Catalysts just speed up that reaction. So this reaction can happen without an enzyme. So you've probably, the person who's commented that is probably thinking amylase, which breaks down starch. It can do it without that enzyme. It's just very, very slow. So your body tends to like to just to use enzymes to speed up the process. Can it be from the OH and the H? It can be from the OH and H. It tends not to be. It tends to be the two AH molecules, which is why you get this strange kink with starch. So I would say almost always, especially for A level, is between two OH groups. Is a bond always C1 and C4 for carbon atoms? So this person is talking about, so 1, 2, 3, 4 in terms of the carbons. They're talking about basically something called a 1,4 glycosidic bond and a 1,6 glycosidic bond. No, it's not always between these carbons. It can be slightly different, and you end up with different structures. Anyone else? Yes, so you can get these bonds happening between different places. I think you do glucose wrong. Ooh. Carbon-free should have OH on the top. No, that's not true. So basically, depending on what type of glucose it is that you're drawing, this can be different. So some glucose molecules are flipped. This little OH group here, it can be up here, but it's not always. That is the difference between alpha and beta glucose. Okay, let's have a look. So people who are a little bit confused, I don't really know what to say. I haven't actually considered what I'm going to say. I think very quickly, if we have a look at it, this bit's easy. I think everyone's got this. The vocab, I think, is straightforward. I think the people who are finding this a bit trickier, it's just the kind of thing you've got to go through. You've got to just be able to draw this again and again and again. And as soon as you've done that, I think you're going to find this quite straightforward. Okay. If not, I'll be here to answer some questions at the end. So I'll move on for now. Those people who've said 2, I'll come back to you. Okay. Right. What I like, in fact, I can try and talk over some bits now. After going through that, let's prove it. Let's prove that you guys are finding this straightforward and all right. Here are 3 little exam questions, which I plucked out of some specification somewhere. Have a go, see if you can answer these. If you can answer these, I think it means you're probably doing quite well. See if you can come up with some answers. I'll read some more comments. Do you need to know how to draw maltose? You don't specifically need to know how to draw maltose. I just think it's important personally. I think it would be useful. So look. Excellent. I've seen one person who has made the mistake of not including one very important word. So a monosaccharide is just made up of one glucose molecule for that person, or one sugar molecule. Lovely. Excellent. I'm seeing lots of people naming the... Thank you for that person who has just said repeating. Yeah, so you were saying that this is a glycosidic bond. Name part A. I'm intrigued to see if you guys can name part A. So I haven't actually told you all the things you need to know. Okay. So we'll have a look at the difference between alpha and beta glucose next. But I'm intrigued. Let's have a look. Question mark. Oh, that one. Yeah. Okay, fine. Right, so I'll go for it. So in terms of what I'm looking for, in terms of what your examiners are looking for, what is a monomer? Most of you are spot on. I'm looking for something that says that it is something that is small. I'm looking for this word specifically, repeating, and then I'm looking for this word, subunit. I think the key word here, the key one which you don't want to not mention, is repeating. Okay, having marked some mocks last year, I think this question possibly came up either a year ago or a couple of years ago. People weren't using this word repeating and they were losing marks for that. Okay, so small repeating subunit. Lovely. Is it subunit or unit? I don't know if I understand that. I've always called it a subunit. It's one word. Do you have to say subunits? Can you say units? I would say subunit. I don't know, maybe I'm being picky of being just having done biology for a long time, you sort of start using the terms that everyone uses. I've always just said subunit. Let's have a look. So name part A. So most of you are telling me that this is a molecule of glucose. Okay. Specifically, this molecule of glucose is actually beta glucose. I haven't actually taught you that yet, but this is beta glucose. Okay. The reason I know that is where these little COC, these little, I'll say it again, cock bonds are forming. You've got one at the top, then one at the bottom, then one at the top, and then you're going to have one at the bottom, one at the top, one at the bottom, one at the top, one at the bottom. That is beta glucose. Okay, you get that structure in cellulose. You don't get that structure in starch. Okay, so you've got one at the top, one at the bottom. That means it's made of beta glucose. Finally, lots of you are telling me the right answer. Bond B is a glycosidic bond. Muy bien. Okay, they are the exam questions. Hopefully you guys are finding that quite nice. Yeah, that molecule would ultimately be cellulose. At the moment, it's hard to say because we can't see the rest of it, but that would be a cellulose molecule. Thank you, Tans. That is very sweet of you. Right, part 2 of the few parts that I've got. So we're going to be looking at alpha and beta glucose. Okay, here are the two, right? See if you can spot the difference. They look very, very, very similar. There is one difference. Have a look, see if you can get it. Do all beta glucose molecules alternate when they bind together to form cellulose? Yes. Beta plus beta. Yeah, so you have one beta molecule, then the other one's flipped, and you have another one, the other one's flipped, it goes on. Guys, what's the difference? Does anyone know the word? So I'm seeing lots of people saying OH switched, the O's or the hose. Does anyone know what the term is for the O's and the hose? Let's have a look. Excellent. I am seeing some people smashing this. You're spot on. So in terms of what you need to know, in terms of the difference between alpha glucose, so this is alpha, this is beta, you need to know that on your alpha glucose, you need to know that the hydroxyl groups, so the OH groups, in case you don't know that, the hydroxyl groups, they are on the bottom, they are on the same side. So both on the bottom. Okay, I'm also going to write same side, which I'll come back to in a second. Okay, so they are both on the same side, they are both on the bottom. You're going to have to try and think of a way to remember that. Okay, something that I always use, I always remember that same side means that it's the thing that makes up starch. Ooh, did not mean to do that. Jesus Christ, how do I get off of that? Let's go and escape. There you go. So same side, that's what starch is made of. I've always thought the little S, I'm not going to touch that again. The S in same is the same as the S in starch. Okay, so alpha glucose, the hydroxyl groups are on the same side. In terms of how you want to define this, in terms of beta glucose, again, this is one of those really, really picky things that examiners seem to like sort of taking marks off of you for. You need to say the hydroxyl group is inverted. So the hydroxyl group is inverted. I have seen examiners taken off marks for saying the hydroxyl group is on different sides. Or if people have said the OH group is on the different side, I've seen them not give that. Okay, so you need to be saying hydroxyl, totally key. You need to say hydroxyl and you need to say inverted. Just saying on different sides won't be enough. Okay. I think that's probably another key bit to this. It's one of those things where you're going to lose marks if you don't say it. So, just because I haven't said this, these are called isomers. Okay, this word isomer, if you've done some chemistry, if you're doing an A-level in chemistry, you might have come across this. Isomer basically means it's got the same constituent molecules, they're just in different places. And I think a nice way of thinking of it is like your hands. They are made of the same things, but look, they don't overlap. They're very different. They're slightly different to each other. If I do that, it's kind of similar, but not quite the same. But that's basically what an isomer is. They have a different arrangement of elements, but it's basically the same molecule. It's still CHO or C6H12O6 in terms of what makes up glucose. Okay, how are you feeling with this then? So have another go at telling me how you are feeling. So that is another really important bit. Let's have a look. How are you feeling? Someone's told me, no, that's chemistry. Unfortunately, you're going to have to remember a little bit of chemistry every now and then. It's okay. So a few of you, I'm seeing a few who are getting a bit confused by this actually. That's okay. Oh no, that person just corrected themselves. Excellent. That's really good, Mark Jones. I'm pleased. Are they optical isomers? In terms of optical isomers, I don't actually know if they ever get called that in biology. I think they just get called isomers. I'm trying to think of what the two different ones are. It's been a long time since I've done my chemistry. I'm honestly not entirely sure. I think they possibly are optical isomers, but I would ask your chemistry teacher or watch our chemistry session tomorrow and ask that teacher because I'm not 100%. I don't want to tell you the wrong thing. Just isomers is absolutely fine. Stereoisomers, that's what I'm thinking. There you go. Someone just said stereoisomers. Yeah, so those of you who are a bit confused, all you've got to really think about is your hydroxyl groups, your little OHs. Go back to my horrible drawing. Here was my horrible drawing earlier. Both of my OH groups here, both of them on the bottom. There's another one. There's another one. All of them on the bottom. That means it is alpha glucose. Okay. In terms of this, going too far. Alpha glucose. Here's another nice way. Alpha. Same. Okay, so the hydroxyl groups, two A's in it. Alpha is on the same. Beta is the other one. Okay, so alpha glucose, both hydroxyl groups are both on the bottom. They're both the same. If it's beta, then they are inverted. Okay, and make sure you use that word inverted. Don't get lazy. I think that comes down to being a big part of this. I think lots of my students in the past have more been lazy than anything else and just sort of thought, oh, I don't need to use that word inverted, which is why I'm explicitly telling you you've got to be using that word, otherwise you will lose some marks. Okay, here's something a bit different. So I've got an exam question here, and these always seem to get thrown in as a bit of a curveball. So essentially, you'll be going along doing your exam, be like, oh, nice, I know this, alpha glucose, la, 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 both on the same side. And then you'll see a question like this and think, oh, God, this is maths all of a sudden. Like, I didn't sign up for this. Here is some maths. I want you to have a go at doing this. See if you can come up with something. So this question is asking you to basically look at some student work. So a student has made a dilution series. Okay, they've used some maltose, which is a sugar solution. He's plotting a calibration curve. And he has a stock solution. So that means his starting solution. So here is his little stock solution in some form of conical flask. And that is 0.6 mole per dm minus 3, which means that there is 0.6 moles, which is just an amount, in case you forgot from chemistry, per every decimeter per 1,000 centimeters cubed of water. So he's got 0.6 mole per decimeter cubed, and he's made some dilutions between 0.1 and 0.6. Complete the table below by giving all headings. So you need to fill this in. So that one, sorry. All the units, so what is the unit that we use for concentrations, and the concentration of the maltose solution produced. So who wants to have a go at working this out? Yes, 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 yes. I can see people saying, no, it's chemistry. It is a biology question. Okay, let's see. Is anyone commenting on how to do this? Excellent. Oh, right. Someone has written in the right concentration. I like that. So this one here is mole per dm minus 3. Okay. This is something interesting. You see this little dash here. If you don't do that dash, you'll lose a mark. If you don't do the dash, you'll lose a mark. You can put it in brackets if you want to. And I have told my students in the past to do both the dash and brackets, but you will lose a mark if you don't do that dash. Did anyone work out what this one's talking about? So if I've got some maltose, what do I need to mix that with to dilute it? Let's have a look. I'm seeing one person who possibly is talking about the right thing, but I'm not 100% sure. Oh, excellent. Bella Marie, I'm going to take your answer. So this one, I'm talking about water. So somewhere over here, I'm going to write water. What's the term that I need to write before I write water? See if anyone can get that. What's the term? Distilled, could be distilled. Yeah, true. I tend not to write it because water implies that it's pure. There's something else that I want you to be saying. So look at how they've worded this. Volume of 0.6 mole per decimeter cubed of maltose solution. Excellent. Jorge has just said it. So I want to say volume of water You probably wouldn't lose them after saying distilled. And then I need my unit centimeters cubed. Lovely. Right, so final part of this then is we need to work out what the concentration would be if I've got 5 centimeters cubed of maltose, 10 centimeters cubed of water, and what that makes if I am starting off with a 0.6 mole per decimeter cubed solution. Okay, so in terms of how you work this out, I'm just going to kind of tell you how to do it. I'm not going to wait for lots of people to say it right and wrong. What you basically need to do is you need to firstly work out how much of this overall solution is my maltose. So I know that 5 units of it are maltose out of a possible 15. So 5/15ths of it is maltose. Okay, I would always tend to turn this into a decimal just because that's, I think, how my brain works. I don't like all these fractions. So that equals 0.33%. Okay, so out of my little pot that I have just made, 0.33% of it is going to be maltose. Okay, in terms of then working out what my solution is going to be, I've started off with 0.6 mole per decimeter cubed. I don't know if you can see that little red thing moving around. I've started off with 0.6 mole per decimeter cubed of my stock solution. So you need to just basically finish this by doing 0.33 multiplied by 0.6. When you do that, let's check my maths. Excellent. You should get 0.2. Okay, so in this question, the concentration of maltose solution I've got is 0.2 moles per decimeter cubed. That's because 5/15ths is 0.33. I guess that should be reoccurring before anyone tells me off. 0.33 reoccurring. If you multiply that by your original, so there you go, here was my original somewhere up here, times that by original, you end up with 0.2. Excellent. I don't know if lots of you are saying that. Hopefully that's not too complicated. If that is tricky, that question, you'll see lots of them. So I would always say, in terms of getting marks, always work out how much you've got. So you've got 5 out of 15 because 5 plus 10 equals 15. That's 0.33. If you started off with 0.6, then you just times that number. I will endeavor to put one of these questions in one of my other sessions at some point, just so you can have another go, because these are the sort of things that you get used to by just doing it again and again. Okay, final part, nice and easy bit. Cellulose and starch. This is the key biological bit, okay? So in terms of what you need to know, I possibly said this was hard earlier, I can't actually remember. I tend to flip-flop a bit. In terms of what you need to know is starch has a function. The function of starch is to store energy. Okay, so I'm gonna write starch is for energy storage. Okay, that is its function. Okay, in terms of linking its structure to that, it's actually made of 2 different molecules. So starch can be made of this molecule here, which is called amylose, and it can be made of this molecule here, which is amylopectin. Just so you know, starch is the one which is in plants. By the way, humans and every other animal have something called glycogen, which is very, very similar, slightly different. It looks more like amylopectin. Okay, so its structure, you need to know that it is made of branched amylopectin. And then there's a really good word for when something isn't branched. I don't know if anyone's going to be able to work this out. Oh, my laptop's dying. Which is linear. So we can say that it is linear. Okay, so we've got a nice linear molecule of amylose. I'm slightly terrified that my laptop is going to die, so I'm going to possibly have to go get my charger in a second. Okay, so that is our structure. So if in terms of starch, it is basically made of either. So you'll have bits of both. You'll have some bits of amylose, some bits of amylopectin, depending on where it is. Plug it in, charge, please. Hang on, bear with me. I'm going to very quickly run away and get my charger and plug it back in. Okay, bear with me for literally 2 seconds. Ooh. Need my charger, it's dying. Where is it? Ah, I'm back. I've got the charger. Sorry about that. That's not the best. Just going to whack that in very quickly. Right, sorry about that. For all of those people who have just wondered where I went, that never happened. Okay, I never actually left, and you never saw that happen. That would have been very embarrassing if it had died. Apologies for that. I will not do that again. I'll make sure I've got my charger, and I'll move that this way. But right, let's get on again. So in terms of starch, that is the key bit. We need to know it's for energy storage. It's made of branched amylopectin, which is this guy on the right, and it is made of linear amylose. Okay. In terms of starch, in terms of its structure, does anyone remember what type of glucose that it has? Anyone guessing this? In terms of Mark Jones, I will get to your question about the properties in 1 second. Excellent, loving it. Yes, amazing. Right, so alpha glucose is the one that makes up starch. Okay, so alpha glucose is the one that we need to know. Okay, so linking the function and the structure is for energy storage. The structure is it's made of branched amylopectin. I'll talk about that in a second, whoever just told me about properties. And it's made of linear amylose. Okay, in terms of its properties, this is where it gets a little bit more complicated. So due to its structure, it can basically wind up in a really, really, really tight little coil. It's almost kind of similar to DNA in the sense that it wraps up into a very, very tight coil. Being compact is really, really beneficial for a plant. Okay, being compact means that it will fit inside of a cell. That is fundamentally important. If you couldn't do that, then you would struggle to be able to use it as an energy storage molecule. Same with DNA. I think I've read somewhere that most of our cells have an equivalent of about 5 meters of DNA and it needs to be wrapped up, otherwise it can't be used. So it's got to be compact, it's got to be able to fit inside the cell, and therefore it can't take up too much space. Okay, this next one, starch. This is, so I tend to see this question awful lot. You've got to be able to tell me about how its structure leads to a functional property of it being insoluble. So why is it so useful that starch is insoluble? What would happen if starch wasn't insoluble? Can anyone tell me this? Excellent. Tans Rex has just saved the day. By being insoluble, and here's a word that you possibly may not have come across yet if you have just started your A-level. Being insoluble, it means it doesn't affect water potential. In terms of what that means, you've come across osmosis, I would have thought. Osmosis is a movement of water from a high concentration to a low concentration. Okay. If you had this molecule, and you have lots of it, if you had this molecule in, it would cause water to move about everywhere. And that's shocking because water can cause something called osmotic lysis. It can literally cause a cell to burst because it's gained so much water. Okay. So, I didn't write the word effect. So it's insoluble. It doesn't affect water potential and therefore can't cause osmotic lysis. Okay, I would learn that off by heart. So starch is insoluble. It doesn't affect water potential. It can't cause osmotic lysis. Okay, the final part. So this part about me talking about amylopectin being highly branched. Right. Someone mentioned earlier that you just need a bit of water to break these little bonds. Okay. Hopefully you're remembering what kind of bonds I'm talking about as well. Glycosidic bonds. You could use just water, but we have these little enzymes as well. You guys probably remember that enzymes look a little bit like Pac-Man, or I always have to me anyway. But if I've got amylopectin, if I've got a branch structure, my little enzyme can come and attack here. It could come and attack here. It could come and attack somewhere down here. It could come and attack somewhere down here. I say attack, I'm being a little bit, well, cute really. When I say attack, I mean it could hydrolyze the amylopectin. But by having branched chains of amylopectin, it means that it has a very large surface area, another term you're going to come across a lot in biology. It's got a very large surface area, so it can hydrolyze, or it can be hydrolyzed quickly by enzymes, releasing energy. Okay, they are the functional properties. So if you ever get a question asking about, I'll come back to Holly in 1 second. If you ever get a question asking about how the functions of starch relate to its structure, you need to be talking about these things. You need to say it's an energy storage molecule. It's made of branched amylopectin and amylose. This means that it is compact, it can fit inside a cell and doesn't take up too much space. It's insoluble, it won't affect the water potential. Osmotic lysis, for the person who asked a second ago, basically, if you are a little cell, if you've got not a lot of water inside you, and we use a little symbol for that, which is a psi. So if you've got not a lot of water inside of you, and you get put in a solution which has a lot of water, there you go, little psi symbol, that just means water potential. Water will move in, so water goes from high to low. If you have too much of something, if you have too much water outside, your cell could burst, right? If this molecule was soluble, which means it dissolved, it would be decreasing your water potential, which is why it could cause osmotic lysis. Okay, that's all you need to know about starch. In terms of cellulose, cellulose is very, very different. So cellulose, you form these long straight chains. So the function of it, and you've probably heard this before in GCSE, the function is it's used in cell walls. Okay, it's very, very strong. It's used in, sorry, not cell walls, in cell membranes. No, I'm talking nonsense again. It's used in cell walls, sorry, my bad. Okay, so the function is it is used in cell walls. That's what cellulose is there for. Not every organism. Some organisms use something called chitin, which you possibly have heard of. Bacteria use something called peptidoglycan. But cellulose is used in the cell wall of a plant. Okay. In terms of its structure, it is made of long, I'm going to say unbranched here, just to make sure we've got the difference. Unbranched chains of beta glucose. Okay, so long unbranched chains of beta glucose. And it's got one other little trick up its sleeve. Because of all these little hydrogens in our carbohydrate, so going back to our structure of carbon, so I've got a C there and O there. Again, you need to make sure you can draw this just like I can, just as quickly. In terms of being beta glucose, let's have a hydroxyl group there. Let's have a hydroxyl group there. Just finish it off like that. And then CH2OH. I'm not bothering to draw the other hydrogens. We can form hydrogen bonds. So you possibly have heard of this at GCSE, but hydrogen bonds are little bonds that you get. They're little forces of attraction that can join up between hydrogens and oxygens. So say I've got another molecule up here, which I'm not going to draw, another molecule of glucose. You can form a little hydrogen bond between the O here and what would be the constituent H of the previous chain. Okay, so imagine there's a little hydrogen here. There you go. Imagine there's a little OH here. You can form a little hydrogen bond sort of between these two here. Okay, so you need to know it's a long unbranched chain of beta glucose. Let's say it is interlinked by hydrogen bonds. And it forms a structure which I haven't actually mentioned, which is called a microfibril. Okay, so cellulose, it seems complicated, but there's not actually as much to remember here. You just need to know it's long, unbranched, has hydrogen bonds, and this forms these big old structures called microfibrils. It basically gives it a massive tensile strength. Okay. Is it true that the cellulose cell will prevent the cell from bursting and it keeps it turgid, increasing the surface area of cells photosynthesis? Yes, so the cell wall does prevent it from bursting. The turgidity, which basically means, imagine a water bomb, like imagine being able to squish it. Turgidity is how filled with water it is. That's not so much caused by cellulose, that's more caused by the vacuole, which contains water, for the person who's just asked that. Okay, so that is looking at cellulose. In terms of kind of giving you the similar sort of properties, the long unbranched chains, it's mostly doing the same thing. This provides strength. In terms of it being insoluble, exactly the same as before. So insoluble does not affect water potential. Okay, so it does not affect water potential, no osmotic lysis. Again, you sort of have to get into the habit of just saying these things again and again and again. And hydrogen bonds forming between the microfibrils. Basically, we need to say this also provides strength. Okay, cellulose is very basic. It's sort of just a molecule to keep stuff strong. And by stuff, I mean cell walls. Okay, so I think for the final time today then, and I'm going to need to stop quite soon because I'm going way over. Let's see how confident you guys are feeling with this. Let's see. So 1 for straightforward, 2 for all right, 3 for super confused. I'll try and answer some questions. So let's have a look. Why does cellulose have beta glucose? It needs to have those inverted glucose molecules just so we can form these hydrogen bonds, and so it forms this sort of structure which is long and straight. Okay, if you don't, it sort of coils up in a similar way to how starch did. Maths question. This isn't a maths question. Yeah, fair enough. The person who's telling me that it's unreactive, that is the case. It is unreactive. Yeah, good point. Am I going to do glycogen? No, I'm probably not going to talk about glycogen. Glycogen just looks exactly the same as amylopectin. Basically, it's animal amylopectin. Why are unbranched chains stronger than branched chains? Because you can basically form these, it's because they're long, essentially. So these 2 points here, they're sort of long unbranched chains. It goes together with hydrogen bonds. By being long and unbranched, they get really, really long. That doesn't make any sense. They're basically longer so they can form more hydrogen bonds, which is what gives it strength. So to be honest, these 2 points are kind of linked together. You kind of intrinsically need to have both of them to be able to understand it. That's a really good question. Is cellulose polar? No, cellulose is non-polar. Let's have a look. How do you revise math skills in biology? Practice makes perfect in my mind. What are microfibrils? Lots of cellulose molecules stuck together. Is the spelling going to matter? I would say at A level, probably. I think if you spell something a little bit wrong, like if you spell cellulose maybe with an O there as opposed to a U, I think if it's clear that you know what you're talking about, it's probably fine. But if you're talking about lots of words and spelling them horrendously, examiners might not know what you're talking about. Excellent. I'm fairly happy with this. Gone. So let's have a go with one last little question for you to have a go at, just because this one always catches people out. So explain the difference in the structure of the starch molecule and the cellulose molecule shown in the diagram above. So be very careful with this word structure. So explain the difference in the structure of starch and cellulose. See if you can work stuff out. Let's have a go. Yeah, good point, Mark Jones. Don't spell mitosis and meiosis wrong. It doesn't have to be polar to have hydrogen bonds. It can be electronegative for the person who's just said that. Yeah, I am going to finish quite soon, guys. For those of you who are saying, oh God, how is this still going on? I am going to finish soon. I apologize for my laptop slightly dying. Lovely. I'm seeing people saying the right thing. This is lovely. Explain the difference in the structure of the starch molecule and cellulose molecule. For this answer, it's actually really straightforward. You need to say that starch is made of alpha. So I'm going to write this in a really lazy way. So starch is made of alpha. You need to be able to say, to get these 2 marks, that the hydrogen bond, sorry, the OH groups, the hydroxyl groups are on the same side. One issue is lots of people will then give up. If you give up at this point, you won't get any marks. So you need to say cellulose is made of beta. And then after that, you need to say hydroxyl groups are inverted. Hydroxyl groups inverted. Lovely. That is all you'd need to say for this question to get those 2 marks. Okay, so hopefully coming to the end then, you now know what I mean in terms of what a monomer is. Nice repeating subunit compared to a polymer. You know that alpha where we've got the A, so we've got the hydroxyl groups on the same side, differs to beta, and hopefully we know how cellulose and starch relate to their function, their structures relate to their function, including their properties. They have key things that I need you to know. Okay, just to show you a few cool things that we've got. So in case you were wondering what this sort of session was, this is SnapRevise 2.0, what you are seeing in front of you. This is me being part of your tutor to help you get through these tricky few months, or these tricky few years, really. This arrives on October the 16th. Okay, so you want to be keeping an eye on this. There is SnapRevise at the moment, which doesn't include this bit that you've got right now, but I'll talk more about that in a second. Okay, just so you can have a look. Ooh, apparently I can discard my ink. That's fantastic. I'll show you what the website looks like just so you can see this. Whoa, this is all going crazy. Let's have a look. Not that one. Here we go. Lovely. Hopefully you can still see this. Can we still see this? I'm going to scroll up to just check that you guys can see what I can see. Oh, apparently there's a bit of a lag. Let's have a look. So in terms of SnapRevise 2.0, this is what we are offering you. So the way it works, just so you can like get the, well, you can see that you're getting your money's worth, is the way it starts is you pick a topic. So this is meant to be really personalized, and it uses some really smart tech to make sure that you guys are getting exactly what you need. It starts with a subject. So say you are picking the lungs, okay? After you do that, it'll do a little diagnostic test. It's a great word that's educators like to throw around. It'll do a test to check what you know. Okay, this person here who's done it, look, cool, I didn't know the alveoli and the bronchioles, rubbish. Following that, look, you can see that you'll be given some personalized learning paths. This is why I said it was smart. It knows what bit you didn't know. Look, it covers them in yellow, the bits you don't know, and it will get you to watch them. It will then give you a whole load of questions. This is what's great about SnapRevise. You have literally thousands of questions for you to have a go at doing, which you can work through at your own pace, preferably not in lessons or anything, but you could go through it. You could check your understanding. Our lovely people at SnapRevise have also made these beautifully designed little revision posters. So you can stick these up wherever you want. And I would really recommend you do this and put them in awkward places where you have to read them every day. But yeah, here's something about enzymes, for instance, that you could print off. Covers the spec points too, which is pretty epic. There are some walkthrough videos that we can offer you. So here is an exam question that maybe you wouldn't know how to answer. And we'll go through like the command words, so the explain, describe, compare, whatever. It'll tell you what you're looking at, and then I'll give you some context at the top. Here are some more exam packs that I mentioned earlier. Look, this is cool. So you can have a look at the easy questions, the medium, or the hard ones. So maybe you're not finding the bronchioles very easy. Maybe you want to do some easy questions, but maybe something, I don't know, carbohydrates, for instance, you're finding an absolute doddle. You could do some hard questions to sort of work on it. Of course, it is gamified, so you can get a little progress tracker telling you how well you're doing, so you can compete with your mates. And then finally, you have me, essentially, who can answer some of your questions whenever you want us to. Okay, it's meant to be interactive. It's meant to be a slightly more, I don't know, a slightly more intelligent way of you guys doing your A-levels. I wish I had something like this. I just had books when I was doing my A-levels, and that wasn't particularly exciting. But that's what we've got to offer you in terms of SnapRevise 2.0. Oh, just so you know as well, some of you were mentioning chemistry earlier. Look, if you want to have a look at our other videos that we're doing, I'm going to finish very soon. Before I give you that code, don't you run away just yet. If you go on YouTube, sorry, and go on SnapRevise, if you scroll down, you'll see here are our upcoming live streams. Here's one I'm doing right now. Here's a chemistry one, which is tomorrow. I'm doing a photosynthesis one for year 13s tomorrow, on Wednesday, which you guys can tag along to if you want to. If you're year 12, I probably wouldn't recommend it unless you really want to get ahead of your class, in which case fair play. And then some more chemistry coming up in the future. So we've got 4 weeks of this. So last few things then. If you were hanging around just for those last few minutes to get this code, here is the code that I mentioned earlier. If you want to get money off, and this isn't SnapRevise 2.0, this isn't this service that you've got right this second, just because we haven't launched it yet. If you want to get some money off all of those things that or not all of those things, the majority of those things I've just mentioned, so the exam questions, the posters, the videos, then feel free to use this code, which you've got to use before midnight tonight. And hopefully I won't talk too much longer, so it's not midnight when I finish. But look, you can have a massive discount on this, so almost half price. Okay, that'll give you access for 2 years. It's pretty good. It also will give you a discount when you ultimately get SnapRevise 2.0 if you've enjoyed this. If you haven't, then maybe don't. But hopefully lots of you have. Otherwise, I am here for questions. So if you would like to ask me any questions, I can do my best now to go through some of them. So I'll sit here probably for about, I've already gone over by quite a lot. I'll wait around for about 5 minutes. If you would like to ask me any questions, feel free to. Hopefully my team who are answering your questions will also be able to help too. Okay. Oh, I spoke for a long time. You couldn't see anything. What was the code for SnapRevise? Oh dear, sorry. Can you see that? If I go back up again, are you just seeing me? Oh God, this is embarrassing. Hang on. If I go back on here, hopefully you'll be able to see this maybe. Hang on. Hopefully you can see this again. Hang on. If I put that there, can you see that? Finally realizes, oh dear, how embarrassing. We were just seeing you. I can only apologize for that. There you go. Muy bien. So yeah, there is the code if you wanted that. Sorry about that. Question, the table question. So, right, let's go for, so table question. I think that's not quite specific enough just yet. So I won't be able to answer that. Lydia, I thought the cell wall exerts inward pressure to prevent additional influx of water. Yes, true. That is a good point. In terms of, I'm not entirely sure actually what you're trying to question there. You're right in what you're saying. Is this to do with how cellulose and starch are insoluble? I don't know. Sorry. What one is inverted again for that person? That one is beta glucose. Question with moles and the concentration and volume. Okay, hang on. If I go backwards for that one person there, here we go. I've deleted my ink. So you're told here that he has a stock solution of 0.6 moles per decimeter cubed. So he's got a little pot of stuff here, which is 0.6 moles per decimeter cubed. And this person has then used, he's got another pot, and he's taken from here 5 centimeters cubed of maltose, and he's put in 10 centimeters cubed of water. Okay, so you need to know what percentage for concentrate, or not percentage, what concentration this person has just made. Okay, so the way I've done, I always do these questions for whoever it was who just asked that, is you basically know that you've got 5 out of 10, which is a fraction. Sorry, 5 out of 15, what am I on about? Because it's, let's erase that. Where are we? So because I've got 15 in total, it's 5 out of 15 of this beaker here. So here's my little maltose, I'll color it in red for you. 5 out of a total of 15 is maltose. Okay, 5 out of 15 is 0.3 reoccurring. Okay, so 0.333333. So that's what percent of it, of this solution here, is maltose. In terms of working out what the concentration is, if I know that I originally had 0.6 moles per decimeter cubed, then you need to do 0.3 reoccurring multiplied by 0.6. And when you do that, that will give you an answer of 0.2. Hopefully that's helped you with that. Let's have a look. What else are we saying? I am not really helping with this because I think my colleagues are helping answer most of this. Most of you guys are asking questions about the courses, which is lovely. Does anyone have any more biology questions? I think it'd be really funny if I had loads of people now just saying no all at once. No, I'll just sit here and I'll carry on talking about various things. Feel free to mute me. Hang on, I'll leave that there for those of you that wanted to have a look. Oh, lovely. Thank you, Joe Lawrence, for making me actually have something to do. I really appreciate that. Do we just need to know alpha and beta structures? Yes. So unless I was talking earlier that there are some things that I avoided from, I think it was in just Edexcel Salters B, you needed to know the sugar which is in, you need to know the structure of ribose. That was the only other thing that I could think of. So yeah, alpha and beta are the only types of glucose you need to know. Benedict's test, whoa, who just said plants are so boring? Right, Billy Chow, whoever you are, I'll come back to Benedict's test in a second. Plants are great, and I spent loads of my degree doing loads of genetically modifying of plants. So Billy Chow, plants are the best. Benedict's test, sorry, just so you know what that is. Benedict's reagent is basically a way of working out if you have a reducing sugar. So reducing sugar being one which is basically glucose. Essentially, you mix it with Benedict's reagent and essentially you heat it up. And if you've got a reducing sugar present, it goes from a blue color to what is always referred to as a brick red color. Okay, that's pretty much all you really need to know. So Benedict's test is something which will test for glucose, which is a reducing sugar. If you have something which is a disaccharide, if you want to see if fat's present, you firstly have to break that glycosidic bond and use some acid to help do that. What do you say when it asks for properties of carbohydrates? That would be such a vague question, just for properties of carbohydrates. I don't think they'd ever ask it. So you'd need to be able to talk, well, if they did, as an essay maybe, which I don't think they would, you'd have to be able to talk about how cellulose and starch, how their structure relates to their function, so what they look like and what they do, and then be able to talk about them being, I don't know, insoluble, talk about how they have a large surface area if it's starched, if they have a really strong structure due to hydrogen bonds if it's cellulose. Is peptidoglycan the same as murin? Yes, peptidoglycan is the same as murin. It's bacterial cell walls. Properties of carbohydrates, the ones I've just mentioned, whoever that was, it's actually the most boring part of biology, you have to admit. No, Billy Chow, I will not admit that, never. What board do we do? The person who's just asked what board do we do, I'm doing all of them. So I'm putting these sessions together to cover AQA, Edexcel, and OCR. I'm going to take the bits which are in all of them, and I'll make sure I do, well, say 70 or 80% of them are the same, and then I might leave an odd bit here or there, but I'll make sure that you've got links to things which are like the additional bits, such as the ribose sugar. Can you talk about hydrogenation? Hydrogenation quickly, please save my A-level. Hydrogenation? That's chemistry. Who's talking about hydrogenation? Interesting. Hydrogenation, I'm sorry, I don't think that's my expertise. Hang on, let's have a look. So saving your A-level, I feel like I need to. Just going from what Wikipedia says. Wikipedia says hydrogenation converts liquid vegetable oils into semi or solid fats, such as those present in margarine. That's what I thought it was. So hydrogenation, if it's turning something into a solid, then it's going to be removing the double bonds. I don't know if that's helping you, whoever's just asked me that question, but yeah, essentially, if you want to do hydrogenization, it's basically going to be removing double bonds to turn something into a solid. I really hope that's helpful. It might not be. What else? Ester bonds. Whoever has just said ester bonds, Adil, ester doesn't have an H in it, firstly, and we're pedantic about things like that. It's just a bond you have between glycogen and a fatty acid. It's used to make lipids. Reducing sugars are basically sugars that can reduce other things. So you may have come across the word oxidizing and reducing. Reducing basically means it can strip off an oxygen off of something, or you possibly have come across oil rig. Oxidation is the loss of electrons, reduction is the gain. It can cause other things to gain electrons. The examples of reducing sugars are the monosaccharides. Tommy Shelby has just started A-level and it's kind of hard, but not that bad. That is possibly the most interesting thing I've read all day. Teach me phospholipids. Definitely saving you. I'm going to say no, I'm not teaching you phospholipids. You've got to come again next time. Is amylopectin a variation of alpha glucose? Q-ish. No, it kind of spirals up. So no, I wouldn't say that. How does hydrogen bonding occur? Hydrogen bonding, basically, this is going way into more depth than I need to. Hydrogen molecules are very slightly negative, whereas oxygen is very slightly positive, which means they stick together. Isn't ester between glycerol and fatty acids and not glycogen? Yes, sorry, Rancid Flow, you were spot on. Ester is between glycerol and fatty acids, not glycogen, my bad. Oli, in photosynthesis, what's RUBP? Sorry, so RUBP is something which I'll talk about in my next session. It's a little molecule made of 5 carbon atoms. I'll tell you about that in my year 13 session. Biomolecules is the best chapter, hands down. You need to go into biochemistry, whoever that is. Right, I think I'm going to stop this now because I think we've all been here for long enough. So I am now going to stop this session. I'm going to pause my live stream. So I'm going to stop sharing this. So hopefully you can't see my screen anymore, although saying that, apparently I don't really know what I'm doing. Hopefully that is staying there. And then, yeah, this is going to be the end of this session. This is the end of the live stream. So thank you very much for all of those who've come here tonight. I hope you've enjoyed it. Those of you that are left, hopefully it's been really good. And I will see you later.